Welcome to our annual CAA Alumni Panel. I am Freya Karsh, Chair of this year's Columbia Alumni Leaders Weekend Planning Committee. I am really excited to see so many Columbians on hand for the kickoff of our 8th annual Leaders Weekend. This year marks our largest and most ambitious weekend to date. This year's theme, Powered by Networks, is an appropriate description of the evening's activities. The evening's panel, the photo retrospective of the Images of the Brain, the Columbia Alumni Arts League reception, or any of tomorrow's activities. Every part of this weekend is designed to highlight the power of Columbia's alumni network. We are extremely excited with the topic this year, Understanding Our Brains, Understanding Ourselves. Further, we're extremely honored to have such an illustrious panel of Columbia alumni experts. This event marks the start of the Columbia Alumni Weekend. Joining us this evening are many alumni leaders from around the globe. Tomorrow, many of you will participate in the Leaders Conference, and still others of you have already participated in school-based meetings or regional and affinity training. The efforts on behalf of Columbia and your fellow Columbians is greatly appreciated. I want to thank the members of our planning committee for their insights and work during this process. On behalf of the CAA, I would like to thank Carl Schoonover, a doctoral student in neurobiology at Columbia University, for his visually arresting images of the photoretrospective from his book, Portraits of the Mind, Visualizing the Brain from Antiquity to the 21st Century. Carl's book will be available tomorrow at CAA Alumni Authors Book Fair in Alfred Lerner Hall. I want to take a moment and recognize my fellow members of the CAA Board of Directors. Please stand. Thank you, all of you. Also, we have a number of distinguished Columbia faculty in the audience this evening. Again, if they could rise, we would like to recognize you. Please stand. It is now my pleasure to introduce tonight's moderator, Pulitzer Prize winner Jonathan Weiner, the Maxwell M. Geffen Professor of Medical and Scientific Journalism. Jonathan.
Thank you very much, Freya. Is the sound all right? Thank you to everyone in the CAA organizing committee for pulling together this wonderful event. Special thanks to Freya Karsh for your introduction, and to my cousins Janet and Michael Kornfeld, who are here in the front row. And what a treat to discover that some of the organizers of this event would be hanging out in the green room with us, and we go way, way back. So it's very, very nice. I have to say I'm here slightly under false pretenses, very slightly, because if you look in the program and you see the P after my name, that means that my degree is parent. I have a son who graduated from Columbia undergrad 2 years ago, but I also have a father who came here, got his PhD here in engineering, taught here for decades. I sort of grew up toddling around on the low library steps, and I've been here for 7 years now as a professor at the Columbia Journalism School, teaching graduate students how to write about science, nature, and medicine. So this program, as many of us already know, is presented in conjunction with Brain Month, a program of the Mind Brain Behavior Initiative, and we'll be looking at these kinds of issues throughout the month. As part of it, I'm looking forward to conducting an interview with Eric Kandel, which you can watch on the web on Columbia Giving Day, Wednesday, October 24th at noon. So these are the kinds of cross-disciplinary conversations that are just going to be hardwired into Columbia in coming years, thanks to the new campus taking shape in Manhattanville, where the Jerome L. Green Science Center is being built as a new home for the Mind Brain Behavior Initiative, MMBI. And it's going to be very, very exciting to all of us to watch this take shape, to watch this happen. It's going to be bringing together great neuroscientists, biologists, physicists, psychologists, and even artists and writers. 1,000 people are going to be working there. It has a sort of Jules Verne quality when you think about it. And there's a great story about it, which you may already have seen in the fall issue of Columbia Magazine. So when I thought about what to say here in the presence of these panelists introducing this event, I remembered something that a friend of mine asked the first neuroscientist he ever met. It was freshman year of high school. A neuroscientist had come to visit and given a presentation, and afterward my friend Mike Shepard stood up and said, really bemused, almost as if he was talking in his sleep. He was so caught up in the fascination of the subject, which we all feel, and the question on his mind. And he said, what is a thought? And all of us in the audience, or many of us in the audience, started to sort of titter. I mean, remember, this is high school. And it sounded like such a simple-minded question. But really, it's an extremely deep question. And an enormous part of the excitement of this gathering of neuroscientists at this institution at Columbia and building now with this initiative is that it seems as if we are approaching the moment when we can begin to answer that question. An extraordinary explosion of knowledge in the neurosciences over the last decades, and it has brought us to the point where we can begin to ask those fundamental questions that reached each of us as children or young adults. What is a thought? What is a memory? What is a feeling? What is an emotion? All of that we will approach, we will watch these scientists approach through the work that they've been doing to date. And as they come together, it may be that right here at Columbia, those answers will take shape. So I'm very pleased to introduce our distinguished panelists. Richard Axel, who holds many titles, including University Professor at Columbia, Professor of Biochemistry and Molecular Biophysics, and of Pathology at the College of Physicians and Surgeons, investigator of the Howard Hughes Medical Institute, and a Nobelist. I will let Richard, I will let each of the panelists explain the work that they do, but I will say that Richard has given us perhaps our most profound understanding of that very, very basic, almost childish question. How do we sense things? How do we find out what's out there in the world through our brains? And he's done that through study of one of the 5 senses, through the sense of smell. We have Leslie Vosshall here, one of Richard's former students. She's a pioneer also in the study of the sense of smell. She chose Columbia for pioneering reasons also, I understand. She wanted to be part of the first co-ed class here. She's now one of the star scientists at Rockefeller University. And among other things, she's interested in what makes mosquitoes looking for a blood meal hover over your head and choose you. There's a great profile of Leslie in this fall's bulletin of the Howard Hughes Medical Institute, and the reporter mentions that when she arrived at Leslie's apartment, she found Leslie wearing socks all over her body. So maybe she will explain that to us tonight. And we have Lise Elliott, who got her PhD here. studying with Eric Kandel. She's now Associate Professor of Neuroscience at the Chicago Medical School of Rosalind Franklin University of Medicine and Science. And she's the author of 2 books, What's Going On in There? How the Brain and Mind Develop in the First 5 Years of Life, and Pink Brain, Blue Brain, How Small Differences Grow into Troublesome Gaps and What We Can Do About It. I'm right in the middle of reading that book right now, Pink Brain, Blue Brain, and it's excellent. I highly recommend it. And finally, we have Dr. Neil Schneider, who is a physician scientist at the medical school. He's a graduate of Harvard College and of the MD, PhD program here. He completed his graduate work in the laboratory of Richard Axel and his postdoctoral fellowship in the laboratory of Tom Jessel. And he studies how the brain communicates with the muscles in health and disease, particularly in ALS, amyotrophic lateral sclerosis, and sadly there is a Columbia connection there too. ALS, of course, is known as Lou Gehrig's disease. So we have a slightly unusual format in mind for the evening. We're going to, each panelist will take a turn, present maybe some slides, talk for 5 minutes, 6 minutes about the research in progress. And then one of the other panelists will ask a question or two. So that's how the conversation will get rolling. And then after everyone is presented and our questions up here have been asked, there'll be an opportunity. People will walk through the aisles with microphones and pass them around for those of you who want to ask a question yourself. So without further ado, let's start with you, Richard. Please come up.
It's a pleasure to have the opportunity to talk with you this evening. I graduated from Columbia College in 1967 with a degree in genetics, and I'm going to talk to you briefly tonight about perception. Indeed, I've often been asked by many of my molecular colleagues whether neuroscience is an appropriate occupation for a molecular geneticist, and to them I can only reply with a line from my friend Woody Allen, and that is, the brain is my second favorite organ. So, this is not a nose. It is a portrayal by Belgian surrealist René Magritte of his own brain's representation of the external world. It is a vignette of image and reality locked in mutual cancellation. This problem of how the external world is represented in the brain is not simply a creative force in art. It stands at the center of philosophy, psychology, and neuroscience. We are interested in how the external olfactory world is represented in the brain. More generally, we're interested in the astonishing problem of how it is that the external world, which consists of physical properties, wavelengths of light, frequencies of sound, chemical structures that define odors and tastes, which have discrete physical properties, can actually be represented and sensed in a brain which only consists of neurons, nerve cells. And these nerve cells can only do one thing, and that is fire, spike, and they can only do so and vary in 2 parameters, time and space. And this is an astonishing problem. Now, the observation that you take the real world, since that's all that's out there, and actually are capable of representing it by this rather homogeneous set of neurons, immediately tells you that what the brain is doing is abstracting. The brain does not simply recreate the world in the world's own image. It creates the world in a brain image using a language that we do not understand, but we are currently trying to decipher. And I will very briefly try and describe to you how we are trying to sort out how the brain knows what the nose is smelling. Very quickly, and we can talk about this over drinks, what you see here is the epithelium of the nose. The epithelium of the nose consists of a set of neurons on the tips of which reside odor receptors. There are 1,000 of them. 5% of all the genes in your chromosome are dedicated to the recognition of odors. Each of these cells makes one of the 1,000 receptors, and they're disarrayed in your nose, but order is restored as they send a process into the brain, and all the cells that make the same receptor converge on a single point. If there are 1,000 receptors, there are 1,000 points. And this shows you the points for 10 different receptors. It immediately follows that a given odor will activate a collection of these 1,000 receptors, and that in your brain will activate a collection of these 1,000 points, and the quality of an odor can be discerned by the spatial properties of the olfactory signal that comes into this structure. And that indeed is exactly what occurs. One can do imaging of neural activity, and in green you see the point that is lit up by, in a mouse brain by fox urine. Mice don't like fox, and they run away. And in the red, you see the points that are lit up by banana. And in fact, those points are exactly the same. Those points of neural activity are exactly the same in all mice. And so I can, with an experienced eye, an ignorant but experienced eye, look down on this spatial pattern, and I can tell you the nature of the odor that an organism has encountered in the world simply by the spatial pattern of neural activity. That's not bad, but there's a problem. I do this with a sophisticated microscope and my eyes and my brain. And the problem we need to solve is not how I do it by looking down on a map, but how the mouse brain does it, because the mouse brain does not have a microscope in it. It must have a way to read this map. Let's very, very quickly walk through that. It reads the map for the purpose of generating a couple of kinds of behavior: innate behaviors and learned behaviors. Innate behaviors are easy. They're a cop-out. Since the spatial patterns are indeed stereotyped, certain of these spatial patterns will project to regions of the brain. It's called limbic brain, which elicit innate behaviors, and depending upon the position of the activity and the position of the projections, the mouse will either run away, show appetitive behavior, or mate. We get into a problem because most odors, as you all know, are not innate. They are learned. There are virtually no odors in humans that elicit innate behavior that you are aware of. And what we observe is the region of the brain responsible for innate behaviors discards this odor and projects to cortex, a structure called olfactory cortex, in a dispersive, unstructured way which exhibits none of the spatial order that we observed earlier in the brain. In fact, here is the pattern of neural activity in cortex, and what you see is different odors are represented in the very same way, and the best explanation for this is that it's a mess. It's random. Each odor appears to activate a random ensemble of neurons. And I argue that that's good, because if it activated a fixed ensemble of neurons, then you might respond to a given odor in a fixed way. And for most odors, you do not wish to do this. You wish to learn the significance of this odor and respond appropriately. So you've got this random, disordered abstraction of the world in your brain. And one must, through learning, impose meaning. So what I've described to you, however briefly, is the notion that the cognitive brain, that is the olfactory cortex, is representing odors as an abstraction, as a random distribution of neurons. And if the distribution is random, it can have no meaning, and meaning must be imposed by experience to allow the same odor to have a different to elicit a different percept in different individuals. And so what I've described, this abstraction, is not dissimilar to abstract art. Consider, for example, this very famous painting by Michelangelo, Leda and the Swan. You all recall from Humanities that Leda rebuffed the advances of Zeus. Zeus tried to seduce her unsuccessfully, and he finally succeeded by turning himself into a swan. This is a representational image. And Cy Twombly, about 30 years ago, painted an abstraction of Leda and the Swan. And like the piriform cortex, in this abstraction, what Cy Twombly did was to disorder line and form and color completely so that it no longer bears any figurative representation of the Michelangelo painting. But I and many of you can just as easily recognize this Cy Twombly image which sits at MoMA as Leda and the Swan, because we have learned the significance of that abstraction. And the next time you see this, you will know what that is in much the same way that you can recognize Michelangelo. Thank you.
Leslie, do you have a question?
I do have a question. So what Richard has presented should make all of you unable to sleep for the next year. It's a very provocative proposal that this extremely random pattern that you see can be actually interpreted by the brain. So you presented to the mice cadaverine, which is the smell of a rotting corpse. If you close your eyes and imagine the smell of a rotting corpse, butyric acid, the smell of vomit, we all have similar reactions to these very provocative odors. So how, it's very ordered here, then it becomes extremely chaotic and abstract, but the output is quite orderly and there's consensus. So how does the brain solve the problem of the abstraction to make it concrete?
Could you ask another question?
What are you doing this weekend?
You've asked two questions. That is, what you've pointed out is something that I wish not to discuss. And that is, if what I said is true, that is, it's random and individual, how is it that for many odors there are shared perceptual qualities, even though those odors don't elicit innate behaviors? That's question one. Now let's go to question two. So there are two answers to question one. The first is a cop-out, and that is I say that those shared perceptual qualities are mediated by the structured brain, by the limbic brain, and they are the consequence of spatial order. I don't believe that's true, but it's a good answer. The second, more complex answer is that you can, without going into the details, you can model the system so that order emerges from disorder in the cortex, such that each of us can categorize odors as pungent, pleasant, but you can't use those words because those words are learned. So you can, using mathematical data reduction, actually build order into disorder. But we haven't shown that that occurs. The second question was, how is valence, or how is the response to these odors, pleasant, unpleasant, actually imposed on a random ensemble? And we don't know the answer, but we anticipate that it will be imposed in what is called a top-down process by experiential brain. And we know some of the arbiters of pleasure and expected pleasure. They're neurotransmitters, which, and we anticipate that experience will shape how we perceive the world.
Thank you very much, Richard.
Am I
Leslie,
up next?
please.
Thank you so much for coming. I think it's great to see the show of support in the Columbia community. Richard has done foundational work on the sense of smell in the mouse. And although I do wear pantyhose to collect human scents for my mosquito work, I'll tell you briefly what we do with the human sense of smell. So there's about 18 million people in the New York metropolitan area. All 18 million people want to tell us about how good they are at smelling different things. We've invited a few thousand of them to come to 66th and York and smell odors and tell us what they think. We lock them in a room for 3 hours, and they tell us how strong is it, how pleasant is it, what does it remind them of. The first astonishing thing I've learned is that 5% of our subjects are completely blind to all odors, unable to discriminate this vile smell from the bottle of water next to Richard. It would be as if you would reach your 50th birthday and be unaware that you're blind or deaf. So it just tells you that the sense of smell, it's so important for everything about enjoyment of life, but there can be 5% of human beings walking around on Earth completely unaware that they have no sense of smell. This really is an astonishing finding. People will sit there for 3 hours and say, oh, it reminds me of when I was in a meadow as a child bouncing around. 3 hours later, they're still excited to smell, and we look at the data, and they are unable to smell anything. So there's a delusion to the sense of smell, which I didn't anticipate. And so the second thing that I've learned in studying the sense of smell in humans, is that there's an enormous diversity. For those 95% who can smell something, there's an enormous diversity of opinion about what something smells like. So I can ask you, what color is this dot? Most of you who are not colorblind will say it's sort of red. There'll be some consensus. But if I ask you to smell this, what does it smell like?
A little bit like alcohol or mercurochrome.
Sorry, Richard.
I don't smell anything.
Alcohol. Yeah, rubbing alcohol.
I don't smell it very much.
I need someone who can smell. Charles, can you smell androstenone?
She smells.
Aldosterone.
I have to crawl back on
androsterone?
stage. So this, to me, this is an incredibly, incredibly intense odor that smells as if Charles has run a marathon and I've just stuck my nose into his armpit and I'm smelling it. So we heard chemical, we heard nothing. Yep.
I tune it out.
And if I passed around a vial of vanilla, you would get the same, you would get a diversity of answers, and you wouldn't get the answer that you expect. If I say, what color is that? Most of you would say red. So people have a very difficult time connecting the experience of smelling an odor with the semantic actualization of what it actually smells like. So it's an ancient sense, it's a limbic sense, and connecting it to the verbal centers of the brain is extremely difficult. So in surveying a few hundred New Yorkers, we learned that genetics has a big effect on how we sense odors, but culture also has a really big effect. However, we can say, what is the most pleasing sense across all ages and genders and races and marital status? Vanilla is the #1 odor of New Yorkers. So we encounter vanilla in baked goods, we encounter vanilla in perfumes and shower gels, so New Yorkers love vanilla. So what is the least favorite scent of New Yorkers? Isovaleric acid, which is the scent of sweaty feet, sweaty socks. These things sort of make sense. As Richard has mentioned in his beautiful segment, of course, we have pleasing experiences with vanilla. We encounter it in baked goods and cookies and vanilla ice cream, and so we all associate it with good. Sweaty socks generally are not associated with good. Beyond this consensus that cuts across all races and ages and marital status and educational status, there are really incredible differences that segment by age and race and gender. So we can divide New Yorkers by these different segments, and the one I found most fascinating is people over 35 find licorice much more attractive than people under 35. I think because licorice is something that it's kind of an antique scent. You discover it in Pernod and in candies, and it's a scent that young people just don't get. So because they had an experience with licorice 30 years ago, 50 years ago, they find it pleasant. Okay. And so the last thing that we learned, which I think is, I lost out here because I didn't get someone who was a super smeller of this stuff, is that genes play a huge role in how we perceive odors. And so you've all had this sense that if you go into a room and you have cilantro, you can segment the room into the lovers and the haters of cilantro. And you can segment the world into people who love this scent, people who hate it, and people who can't smell it. And so one thing we did a few years ago was try to associate what's going on in the genome of the person. The genome of the person has an effect on which of the several hundred odorant receptors the human nose expresses and how it's perceived. And so I could say that you probably have a mutation in the OR7D4 receptor, which makes you unable to perceive the rich intensity of this odor. So
I'm not surprised.
And so you're one of the lucky ones, because to me it's one of the most repugnant odors on earth. But the point is that culture has an overshadowing effect about how we perceive the world. But we are also genetic creatures, and so there's several hundred odorant receptor genes. So just as there are a few of you who can't perceive this as red because you're missing the receptors that encode red color vision, there are people running around the world missing receptors that makes them find cilantro not disgusting or beautiful. So this is what we do in my lab.
Thanks, Leslie. And who was it who had a question? Richard, you have a question for Leslie? Just one question.
You alluded to the suggestion that there is no language to describe odors, yet your studies require that the participants use a language. So to what extent is this, what is the implication of not having a language to describe an odor?
So going into it, we recognize that if you just ask people to smell 1,000 odor vials and ask them to give you words, nobody will be able to perform the task. So we give them a cheat sheet. We have a very large poster with 150 different adjectives: cat urine, vanilla, sweaty socks, licorice. And so people they have a reference atlas of a lexicon of potential words, imperfect, because it doesn't really, 146 words doesn't really cover the possible semantic richness of what all smells can encode. But it was impossible to do the study unless we give semantic hints to our subjects.
I have one question. What about those socks?
Oh, see, I'm off duty. I'm not wearing pantyhose. So we have to collect human body odor and guinea pig body odor for our mosquito experiments. So I and many of my lab members are usually secretly wearing Dwayne Reed pantyhose on our arms and legs. It's not pretty. And if I just brought a couple hundred pairs, I could have put the audience to work, but didn't think ahead.
Great. Thank you very much, Leslie. Neil.
Well, thanks. I'm happy to be here and to participate in the panel tonight. I am the neurologist on the panel, and I think my role here is to sort of present the clinical perspective on the nervous system. You've heard from Leslie and Richard, and some of the work that I do in my lab is all based on relatively simple systems like the fruit fly and the mouse that we use to study the brain and the circuitry that underlies complex behaviors. And these are really powerful systems to do this. And the studies that come out of labs that focus on these simple animals really, I think, can tell us a lot, both about the principle and the specifics of how our brain works and how the circuits function that underlie very specific behaviors, our behaviors, and make us who and what we are. But the ultimate model, I think, of the human brain is, of course, the human itself. But we're really, as you can imagine, not a very good tractable model. We're not a good experimental model. We can't do the kind of invasive experiments that we do in the fly and in the mouse and even primates. But there's a way around this problem, and that's provided by nature. There's naturally occurring disease, neurological disease, and these diseases lesion the nervous system, and they do that in a variety of ways. And these disorders reveal specific functions of the nervous system, functions that we're usually not aware of, and we take for granted. So this is what neurologists do for the most part. We observe, characterize, and manage these disorders of memory and attention, language, mood, and personality. We look at deficits of motor and sensory function. And these, I think, provide really important insights into the working of our nervous system. So these disorders occur in a variety of ways. They occur as a consequence of stroke when blood supply is compromised to the brain. They occur as a consequence of traumatic injury, head injury, spinal cord injury that results in loss of functions, demyelinating diseases which strip neurons of their insulation so that they don't function. And the problems also occur as a consequence of degeneration, of neurodegeneration, the loss of specific populations of neurons that underlie specific functions, and their loss results in certain syndromes Alzheimer's disease, Parkinson's disease, Huntington's disease, and the disorder that I work on called amyotrophic lateral sclerosis, or ALS. So, as Jonathan mentioned, in this country, ALS is better known as Lou Gehrig's disease, after the Columbia alum and a Yankee Hall of Famer who died of this disease in 1941. And 70 years after Gehrig died, it's still sadly true that we have no medication that significantly alters the natural history of this disease. We're much better at managing patients, taking care of them in important ways that improve the length and the quality of their life, but we have no effective treatment for ALS. This is a disorder that was first described by a very famous French neurologist, Jean-Martin Charcot, who also described multiple sclerosis. In a very famous paper in 1869, he described a lateral sclerosis, a hardening of the lateral columns in the spinal cord, and the figure here from the original paper depicts this. And he also described the amyotrophy, or the wasting of the muscle that's associated with this disease. ALS is essentially a disorder of two neuronal populations. The so-called upper motor neuron, I don't have a pointer, but in the red strip, in the motor cortex of the brain, resides the so-called upper motor neuron that sends an axon, or a wire, down to the lower motor neuron in the brainstem and the spinal cord. And it activates that neuron, which sends a second axon, a wire, out to the muscle to activate the muscle. And this is, of course, a schematic. It's an oversimplification of the motor circuit. But the motor neuron is the major effector of the nervous system. Its activation, all motor function, all movement, depends on the activation of the motor neuron. And so when the lower motor neuron, again, the brainstem or the spinal cord, degenerates, we see muscle atrophy or wasting of the muscle. We see weakness of that muscle, and a symptom and sign that patients often describe first in their disease called fasciculation, or twitching of the muscle. When the upper motor neuron in the motor cortex degenerates, we see overactive tendon reflexes, like the simple knee jerk that your doctor will often elicit in the office. This becomes overactive. We see abnormal reflexes we call clonus, and we see specific signs, Hoffman-Babinski sign listed here, all evidence of a loss of regulation of motor neuron function that occurs as a consequence of this disease. This is an image on the left of a normal spinal cord. This is an autopsy of a patient who died of let's say, natural causes, and you can see these darkly stained neurons on the left are motor neurons in the spinal cord. On the right is an equivalent section through the spinal cord of a patient with ALS, and you can see that the cord is absolutely devastated. The numbers of motor neurons that are left are very few. So when this kind of degeneration happens, these are the motor neurons that innervate our arms and our legs, the muscles of our head and neck. So when these neurons degenerate, we lose the ability to use our hands, use the ability to walk and to stand, and we lose the ability to talk, to chew, and ultimately to swallow and to breathe. And it's for that reason that ALS is a fatal disease. It's the most common motor neuron disease. It affects about 1 or 2 people per 100,000 per year. And because ALS is such a deadly disease, patients live only 3 to 5 years on average. The number of patients who live with ALS at any one time is quite limited. The onset is in the mid-50s on average, and for the most part, ALS is a sporadic disease, so that most of the patients that we see in the clinic have no family history. This occurs out of the blue with no warning. So what causes ALS? Simple answer is we really don't know. There's a lot of toxic environmental causes that have been suggested, viral, infectious, autoimmune, inflammatory causes that have been investigated. None of these is known to cause ALS. The only known cause of ALS is genetic. And this is a list of genes in which mutations have been discovered, mostly in the last 5 to 10 years, to cause ALS. And what's striking, really, about this list is that we have over a dozen genes here, and a mutation in any one of those genes, most of the cases inherited from one or the other parent, causes a neurological syndrome that's characterized by the selective loss of motor neurons. So the question for the field that this list presents is, how is it that all these genes, mutations in any one of these genes, can cause a similar syndrome? How are they related? What are the common mechanisms that relate these? And this is a major focus of the field and of my lab. And I'll say just very briefly that what we do in my lab, mostly working in the mouse, is to manipulate the mouse genome to introduce mutations that cause ALS in humans into the mouse and to create models of the disease that we then can use to find pathways that contribute to motor neuron degeneration and to identify targets that we hope we'll be able to use to develop drugs to help our patients not only with the genetic forms of the disease, which represent only about 10% of the cases, but all patients. Now, I could talk more about my own work. Jonathan asked me, and I'll do this briefly, to talk a little bit about my perspective as a clinician about ALS. What does it mean to me to be an ALS clinician? What are some of the things that I've learned from my patients? This is a big topic, something maybe we can talk about more, but I thought rather than tell you stories, and I have as many as I think I've had patients, I'd read from a couple of my patients that have written about their disease, patients that are more, I think, eloquent than I. One was Dudley Clendenin, who was a very well-known author and journalist who died just this past year of ALS. And what he wrote that really struck me was, he said, "We obsess in this country about how to eat, to dress, and to drink, about finding a job and a mate, about having sex and children, about how to live, but we don't talk about how to die. We act as if facing death weren't one of life's greatest, most absorbing thrills and challenges. Believe me, it is. It is not dull. This is not dull. This is not about one particular disease or even about death. It's about life. And when you know there's not much left, that is a weird blessing of ALS. There is no escape and nothing to do." And he found it liberating. He said, "I don't worry about fatty foods anymore. I don't worry about having enough money to grow old. I'm not going to grow old. I'm having a wonderful time." And I can't say that too many patients have a wonderful time with their disease, but people do find a lot of meaning in their lives, limited as they are with this diagnosis. And I had the great honor of taking care of Tony Judd, who was a university professor at NYU and a very well-known public intellectual. Tony died in 2010. He had the disease for only about 2 years. And I just wanted to read a couple things from Tony. He wrote a lot about his disease, and what he wrote, I think, helps me and helps my patients to understand it a little bit better. He called it a progressive imprisonment without parole. And he said, if you're someone who works with your hands, you're a painter or a plumber or a bus driver, there's nothing you can do but to retire. But because I'm a writer and a teacher, and I've been able to continue for 2 years after the initial diagnosis, and that is what has kept me going, the fact that I could continue. I could dictate, I could think, I could, so to speak, write. I think I'm running out of time. Should I race through?
I guess you should.
Yeah. So Tony wrote a lot more. I'll end maybe with maybe one other quote. He says, "I thought a lot about life after death. I have no idea whether there's a life after death for me, but I am absolutely sure that there is a life after death for the me living in this world." But he said, but as for the living, I guess my view on that is that I'm now very clear that living can be reduced to the business of communication, because I can't move, I can't travel, I can't act in any autonomous way, but I can communicate with people, and they can communicate with me, and as long as that is possible, one is alive. So I guess end on a more hopeful note, which is to say that because of a lot of developments, both in ALS genetics, in molecular medicine, in stem cell technologies, we're now at a point where we can be hopeful, I think, about developing therapies for our patients, and I'm very hopeful, truly, that we'll have something to offer them in the not too distant future.
Thank you very much, Neil. We're running a little over time, so maybe we'll keep this next question and answer brief. But do you have a question for Neil?
Well, I guess sort of the obvious, not being a clinician, how, how you go to the office and face patients every day when the prognosis for all of them. I mean, since you deal exclusively, unlike a lot of neurologists who see a range of disorders, to know that everyone you walk in kind of has this death sentence, and that would be so weighing, I would think.
It's obviously challenging. It's very emotional, for us and obviously for our patients. It's a great privilege, and I sometimes don't have the energy to do it, but you go down, you sit with somebody, you put a face with a name, you begin to hear their story, and it becomes very personal. It becomes something I often feel very privileged to be part of. But yeah,
I suppose especially because the imminence seems to bring out these very deep sides of people that you might not encounter in more mundane disorders.
ALS is such a dramatic disease. I mean, you see changes over weeks and months. Even for a neurologist, I mean, it's just striking. And that's other thing. I'm always amazed at just how fast this thing progresses. I mean, it's really astounding.
Thank you, Neil. Please.
Well, I want to say also what a privilege it is to be back here, and this is going to be quite a change of gears, hopefully to end on a little more fun note. But I'm going to hope to tie this in a little bit with especially some of the things that Richard and Leslie were talking about this whole nature-nurture issue, about what how much of our behavior really is innate. And if we hear for even the sense of smell, which seems so almost reflexive, to discover that even that is learned, I think really ties in nicely with what I hope to tell you about gender, which is that it ain't as hardwired as this idea of brain sex has lately portrayed. So here we are, I would say in the last 15 years or so, the topic of gender, when people have started to delve into the sex differences in the brain, this is what we're getting. I'm focusing on the popular side because I think it has had it's a good example of how a little bit of science can be quite dangerous, and yet it's had a pretty surprising impact on a lot of our culture. I'm particularly interested in its impact on kids and education, but even business schools are now giving courses on gender-based leadership and so on, based on these little horrible fragments of science that are quite distressing. So it all started with this book, Brain Sex, by a geneticist and a journalist. David Jessel's no relation to our wonderful Tom Jessel, but and then amazingly enough, a couple of books, The Female Brain and the Male Brain, which were widely panned by scientists, but have hit the bestseller list. Luann Brizendine ought to know better. She didn't go to Columbia, but she did get her MD at Yale, I think her undergrad at Berkeley, and trained, of course, at Harvard, and maybe that explains all of this. But yeah, I mean, there's all kinds of references in these books, but they don't have any relationship to the claim she makes about the so-called male brain, which is composed of duct tape, and the female brain, which is composed of telephone cords. and then it goes on and on and on, and there's these endless books about why men don't listen and women can't read maps, why men never remember and women never forget. But it really all started, of course, from here, right? This idea of Mars and Venus, that we're from different planets, that men and women have fundamentally different brains and can't communicate. So that's where it started, and for me, I'm more interested in the developmental side of things, and I discovered that when we talk about children, this Mars and Venus idea has really infiltrated the way that parents and teachers are thinking about their children. There's two culprits, if you will, who are propagating this pseudoscience. Leonard Sachs, who's a family physician who dropped his practice to write this book, Why Gender Matters, and he's written a couple more books where, again, you take a little bit of science and you turn it into this big story about hardwired differences between boys and girls. This was just one little paper that tells us that he interprets as telling us that boys can't talk about their feelings because of the part of the brain isn't connected to language in boys. It's really quite fantasy. And then we have Michael Gurian, who's not a scientist or a clinician, but he really likes the brain, and he likes hormones, and that, of course, explains everything about how boys and girls learn differently. And this idea has resonated around the country, the idea that boys and girls learn differently, which if you really look at the neuropsychology of learning, and there's been lots of studies over the years, is completely unsupported by either psychological or neuroscientific data. There are differences between males and females. I'll get to that in a moment. But this idea of categorically different learners and the idea we need to segregate boys and girls, believe it or not, 500 schools are now across the country are doing that. Title IX's been totally revised. I'd love to talk about that more if you're interested. But I want to really focus on what I'm interested in, which is sex differences in the brain and this huge extrapolation. So my own background, I started with a book on brain development, and along the way came across these claims about male-female differences, and I decided on this title, a very poor title, I'll confess right now, Pink Brain, Blue Brain, before I'd done all the research. And I ended up doing, as I was telling Jonathan, 8 years of research, trying to read every paper I could from both neuroscience and psychology. And really, the Germans got a better title, and the translation is, Are They Different? Which is much more accurate without the pink and blue. And so here's, in a synopsis, what I came to conclude in this book. Sex differences in behavior, of course, males and females behave differently, boys and girls behave differently, but generally they're not quite as dramatic as our Mars-Venus notions. Most of these sex differences, and this is where I was interested from a developmental point of view, are actually quite small in infants. This idea that boys are not social at birth and that girls, talk a year earlier. In fact, the difference is about a month. They're quite small, but they grow larger with age. And in terms of this whole nature-nurture issue, I came into this sort of biased for the nurture side because of my training in Eric Kandel's lab, trying to understand how experiencing the environment changes the brain. We know the brain's incredibly plastic, even in the case of odor perception. In terms of gender, the psychologists have understood this, but the neuroscientists are having a hard time appreciating that. And so if you come away with anything today, I want you to appreciate that there are indeed male-female differences, but this idea that there are two different organs, a male brain and a female brain, is quite far from reality. So we have to actually talk numbers a little bit, and this is about the easiest statistic there is. It's called a difference value. You just subtract the means or the peaks of the curves and divide it by the standard deviation. So in contrast to a real difference between men and women, something like height or weight, or even brain size, which correlates quite well with height, weight, heart size, kidney size, all the organs actually are bigger in males and females, quite a lot bigger, a difference value of 2.6, 2.5 standard deviations. In contrast to that, almost every one of our behavioral sex differences is quite small, this D value of 0.35. And the reason we know that is because Janet Hyde at University of Wisconsin-Madison did this meta-analysis of meta-analyses where she found that 3/4 of all of our sex differences are actually smaller. So there's just a lot of overlap in our ability to read emotion in other people, in our math ability, in our verbal ability. We really are not from different planets. You cannot predict based on somebody's gender what their abilities will be. And so rather than Mars and Venus, the better statement might be that women are from North Dakota and men are from South Dakota. It's about the scale of things. So what that tells us is that if there's these small psychological differences, I mean, there has to be a difference in the brain. We don't just think out of thin air. I think we all agree that now. But the actual magnitude of the differences makes it hard to find differences in the brain. And at this point, there really is no clear link, I mean, there's between brain and behavior for any of our well-known sex differences. And even this figure, which I just saw in Eric's new book, is being reproduced all over the place. There's not a lot of evidence that we have these pink areas and blue areas. So what do we know? A common belief is that men tend to use both sides of the brain, and women, or one side of the brain, and women use both. In fact, you have to look at large studies, and that's the problem, is a lot of these myths get extrapolated from one study with 19 males and 19 females, and it's reproduced over and over in the popular media. But if we look at large populations, you see this overlap. Just like with the behavior, males and females are both left dominant. If females used both hemispheres symmetrically, we'd see a curve right there. But instead, I'm just trying to make the point that for our brains, like our behavior, it's a statistical distribution with much more overlap than difference. Here's another huge study, 1,400 subjects from 35 imaging centers around the world. The pink area of the brain is an area that's a little bit more highly connected in women. A blue area is a little bit more highly connected in men, just resting in the scanner. But look at the distributions of these 1,400 subjects. It's mostly overlap. The differences are just small and statistical. They're not categorical. So I just want you to take away that, and also this idea that just because we see a difference in the brain in men or women or any sorts of groups doesn't necessarily mean it's hardwired. So consider, here's a little bit of a thought experiment. 2 subjects lying in the scanner. They're given a test of, in this case, moral judgment. They're supposed to reflect on a scenario of behavior and decide whether or not they behaved in a moral fashion or not. And so subject X, strong activation of the medial prefrontal cortex. Subject Y, this little blip on the frontal pole. I'm sort of deliberately misleading you to think that they're a male and a female, when in fact the subjects only differed in their religious upbringing. So the point is that gender learning, starting with the prenatal ultrasound, because what baby isn't born to parents who know their gender even before they're born now, gender learning is at least as potent as other cultural experience in shaping the way our brains function. So I have made this point in my book, Pink Brain, Blue Brain. And since after my book was published, I'm happy to say a couple other books came out that critique exactly the same science and have made exactly the same point, that this notion of brain sex is quite fallacious. One of them, Rebecca Jordan-Young, is right here at Barnard, so she's a young faculty member. And Cordelia Fine's book, I love the title, Delusions of Gender. She's a neuropsychologist at Melbourne in Australia. So that's the take-home message, the myth of brain sex. Thank you.
Thank you. Thanks very much, Lise. Neil, do you have a question? And then we'll turn things over to you.
Yeah, I mean, I don't know too much about this, but from what I've read, there are known differences, anatomical, physiological differences, even in simple systems like the mouse, between a male and a female. Functional imaging studies between men and women have shown significant differences. Women have different, let's say, susceptibilities to psychiatric disease, different responses to therapies. I mean, I understand the need, maybe to not overemphasize or overplay these differences in development or otherwise, but I just wonder if we, do you agree that there are these differences, and if we don't take them seriously enough, might we, miss opportunities to?
Absolutely. I tried to state from the outset there are anatomical differences, there are functional anatomy differences, there are neurochemical differences, but it's a question of degree. These are small statistical differences, not categorical differences. And even taking things like psychiatric disorders, for example, women are twice as likely to suffer from depression as men are. And yet, people have looked long and hard for hormonal correlates of that and have not been able to find it. And one factor actually is socialization. So when you feel down, when you don't, when life is getting bad, women tend to internalize, whereas men, because they have higher power in society, will do more, be able to, engage in more conduct disorder, or they may turn more to alcoholism or drug abuse, whereas females, it'll be anxiety or depression. So the idea is it may be the same stress triggers, but because of our social status, there are acceptable ways to express these or not. And I think the best example actually of that is probably anorexia nervosa, 10-to-1 ratio. It's been pretty clearly shown that it has to do with the culture that you grow up in and what sorts of things are on the magazines and the television. So.
Thank you very much, Louise. Okay, your turn. Please wait until a member of the staff hands you a microphone before asking your question, and why not tell everyone your Columbia, your name and your Columbia affiliation too. Yes. You're a long way in for the microphone, but I guess it's...
There are about 30 of us, maybe more in the audience, who can't smell, given the 5% rule. How do we know? How do we know that we can't smell? If we can describe smells, how do you know? How is the fantasy different from reality? What if I can't smell?
I don't have an answer. I think it's the most puzzling thing I learned in the last 10 years, how people can have this illusion. As I said, if you're blind, you know it, right? Nobody, maybe with babies, it might take a few years to figure out that the baby is blind or deaf, but you have people who are into their 60s who have never smelled anything. So I think maybe, again, it's just a learned thing that someone will hold up a banana and someone will peel it and say it smells like a banana. And so you somehow get this illusion that you're trained by the visual stimuli that this is good and this is bad, and that you in your mind think that you're smelling it. But it's only if you come into the clinic and we look at the anonymized data that you really aren't. So
Isn't a lot of that acquired, though? I mean, if you've had a lot of respiratory infections and things.
you can lose your sense of smell through many different routes. You can be born without a sense of smell. You can have a car accident that causes an injury. You can have a viral infection. So all of us will lose our sense of smell this winter when we have flu or cold, right, where the nasal epithelium stops working. But I'm most fascinated by people who their whole lives have never smelled anything, and I don't really have an answer. It is puzzling.
But do you know that you haven't smelled anything for your whole life?
Yeah.
You used to smell.
Maybe I still do.
And
Yeah.
maybe you still do.
Yeah.
And as we spoke before, there is no language for smell. So smell is very associative. And that is, you describe smells by virtue of the memory of experience. Like the smell of Madeleine for Proust conjured up 7 volumes of remembrances of things past. And so the act of smelling and the nature of the object can elicit memories of smell, which you report.
Yes.
I have 2 questions, one for Dr. Axel.
Please tell us your name and how you came through Columbia.
Well, my name is Stephen Mandel. I'm a neurologist. My wife's the Columbia person.
Great, thanks.
I'm the invited guest. So two questions. One to Dr. Axel. Why is loss of taste and smell an early predictor of Parkinson's disease? And number two, for Dr. Schneider, could you clarify the role of repeated concussions and the similarities in the brain that have recently been reported in ALS?
Could you ask both questions to Schneider?
I could try that one, Richard.
Yeah.
So you know, the first question I'm going to take for Richard. I'm going to help him out here. These disorders are selective disorders, right? Parkinson's disease is a movement disorder. Yet we know actually, I would have said if you'd asked that one of the earliest deficits in Parkinson's patients is sleep, right? So the selectivity of these diseases for the motor system, cortical system of any, is relative, right? I mean, we see the thing that's most obvious. You see the tremor, you see the bradykinesia in Parkinson's, and you don't necessarily see the thing that's there first, and the selectivity is, again, relative. And the same is true, similar, I guess, answer to your question about traumatic brain injury. There was a New York Times piece, maybe you're referring to last year, did Lou Gehrig have Lou Gehrig's disease? And this was a reference to a study that was done out of BU in the VA system in Boston, where people, where these pathologists found a kind of pathology associated with TDP-43 in the brains of patients with traumatic repeated concussive injuries, boxers, football players. And they found TDP-43 pathology, which has been found in ALS patients. In fact, a mutation in TDP-43 causes ALS in some families. And so they said, well, maybe Lou Gehrig didn't have ALS. Maybe he was beamed too many times by the ball, which he was, and that he had a TDP-43 opathy that was not ALS. The simple point, I'm going on too long, but is that these pathologies cross over, right? And these diseases are not quite the silos that we once thought them to be. And there's pathological, clinical, genetic evidence that links ALS to frontotemporal dementia, to chronic traumatic encephalopathy, to these other disorders that probably involve common pathways. And whether you get FTD or ALS or something else, depends on factors that we don't yet understand.
Excuse me. Could just start?
I'm
Okay.
Alex
I have a question here.
Oh.
I'm up here in the top, looking to your right, and I'm waving the thing. And I am asking the question of you that just waved back to me. Thank you. Just in terms of the statistical importance of these differences, and you were saying that it was low, but if you look, for example, and it's not related necessarily, but historically, genetically, we used to think that, for example, cats and dogs were completely different, and we now know that they're quite close. So what determines statistical importance in these differences? How do you determine that these small differences are not statistically significant? I mean, what's your basis of making that decision?
Yeah. Well, every grad student gets to read How to Lie with Statistics along the way, but and that's why, to say something's statistically significant, if you have a small sample, you need a huge difference. If you have an enormous sample, you can get a statistical difference with a tiny difference, and that's why I focus on the D value. It tells you how big are the differences. And I was hoping that those curves would illustrate the degree to which something overlaps or doesn't. And I flashed by a slide where we were looking at lateralization differences between males and females. But another way to think about this is how much of the variance is explained by sex or gender. So by variance, we mean, there's a distribution across the population. And in the case of brain lateralization, who uses their left hemisphere more than the other? Gender only accounts for about 1% of the variance in brain lateralization. So I think that's a nice way of telling you, the degree to which something is influenced by a certain factor, how much of the variance is explained. And that's what we tend to do in nature-nurture analyses, how much of the variance is explained by genes versus environment. For IQ, it's like 50-50. So hopefully that might be a better way to think about it.
And now it's your turn.
So my name is Ali Ashrafi. I'm a civil engineering graduate from Columbia. My question is for Dr. Axel. You showed that different smells create kind of random looking patterns in the brain, right? Now, that random looking pattern, would it be the same if you looked at two different mouses, or it would be actually different in each animal? If it's the same, it's kind of like my vanilla is your vanilla. If it's different, maybe it's really each vanilla is different. Each red is different.
Yeah, you cause me to emphasize an important point. That is, the extent to which we can discern pattern, and when you don't find it, you don't ever really know it's not there. But the extent to which we cannot discern pattern seems to indicate that the same odor will elicit a different distribution of unpatterned neurons in different animals. The implication being that these patterns can have no innate meaning. That's a stretch, but my argument.
My name is Jackie Cohen, and my partner is a Columbia alumnus. And this is a question directed to Leslie. I'm going to indulge myself because this area is so fascinating to me, and ask a question based on personal experience. When I was a child, my life was dominated by smell. I chose my friends and the people I couldn't go near because of what almost appeared visual. The smell experience was so powerful. And I do want to comment that if some adult had asked me about it, I did not have the language to talk about it. Things were either they smelled good or they didn't. And that was sort of the end of it, or I wanted to smell more, or I was repelled. So what I've noticed as I, have progressed in my adulthood is that reliance on smell, that awareness of smell has diminished. And my reliance on my other senses have come to the fore. And I guess I wanted to ask you if you could talk a bit about what happens to smell as one develops into adulthood.
Interesting question. And I have to say that's going to have to be our last question. So
I'll answer it
Leslie,
quickly.
you're going to wrap up for us.
So we would have loved to have recruited you to our hospital when you were 8 years old, but maybe some smart people in the audience can set back time. So a lot of things happen through puberty. There's really enormous changes that happen in the brain. That's one thing, and then bad things happen when we age, right? So, there's just a long, slow decline to death, and and so all of the senses.
Don't sugarcoat it.
All of our senses decline with age, some more rapidly than others. So for sure, if we rank our several hundred subjects into those who are younger than 35 or over than 35, there's a huge disparity. So people over 35 have a significantly worse sense of smell, either due to environmental insults or other things, or cumulative viral infections. So definitely the sense of smell, like every other sense, becomes less acute with age. Why you had this intense relationship with the olfactory world as a child and why that diminished, I don't have an answer, but it's a fascinating story.
Thank you very much. Thanks to all of our panelists.
My name is Brian Chrisberg, college, class of 81, law school, class of 84, former president of the College Alumni Association, member of the College Board of Visitors, and the vice chair of the University Alumni Association. On behalf of the CAA, I would like to thank Richard Axel, Lise Elliott, Neil Schneider, and Leslie Vosshall for sharing their expertise with us and for making it accessible to all of us. I would also like to thank Jonathan Weiner for moderating this evening's wonderful discussion. Please join me in showing our thanks for an exceptional evening. To echo Freya's remarks at the opening of the program, this evening, and in fact all that CAA does to engage our alumni in the life of the university and with one another is made possible by the power of networks. I strongly encourage each of you to expand your alumni networks outside in the lobby this evening. Before I close, I want to remind everyone that tomorrow there are 2 exciting opportunities open to all alumni. At 2:30 p.m., we invite you all to Alfred Lerner Hall for the first CAA Alumni Book Fair. There will be over 40 alumni authors from nearly every school on hand to speak about their current publications. Following that, at 3:15 p.m., there will be a special Manhattanville briefing with Executive Vice President Robert Kasdan and Provost John Coatsworth. We welcome all of you to join us on campus tomorrow in Lerner Hall. I've also been asked to mention 8 days from today, on October 20, is homecoming. The Lions take on Dartmouth at 1:30. There's a soccer game later in the day. There's a Big Tent barbecue and a family carnival at 11 a.m. at the Baker Athletic Complex on West 218th Street. The CAA finally would like to invite all of you to attend the Columbia Alumni Arts League. I'm told that's pronounced Cal. A reception in the lobby. Please enjoy it. Those of you attending the Leaders Weekend, later this evening there is late night at the Columbia Club of New York, and a bus will depart from here at 9 p.m. to take you down there and carry on. Again, thanks to our panelists and our moderators for a wonderful evening. Good night.